So this semester, I've been fortunate enough to work on a research project in my molecular biology course taught by Dr. Janine O'Brien. And in this, we were looking for bacteria in snow samples collected from her backyard. And our main objectives of the project were to identify the presence or absence of 16S bacterial genes in our snow samples from the 23A storm. We then were trying to identify the presence or absence of INP genes, such as INAQ, in snow samples to determine if snow nucleating bacteria is present. And the third one was to compare the abundance across samples for the 16S genes to determine if various species of bacteria are present. So a little background information is bacteria is everywhere. It's in the air, it's on the ground, it's up in the clouds, in the sky, and snowflake particles. Snowflakes can consist of water and small particles, such as dust or bacteria. And how snowflakes form is water vapor droplets condense on the particles in the atmosphere, such as the dust or the bacteria. And it freezes, which can cause this branching formation on the water vapor on the particles, as shown in figure 1A. And as more and more water vapor condenses onto the molecule and the heavier the snowflake becomes, it ends up falling down to the ground. And certain types of bacteria, not all bacteria do this, but certain types of bacteria has these, such as P. syringae, has kind of mutated over time to possess these proteins that allow this nucleation to occur, which would help the bacteria travel. So, winter storms has a tendency to move from west to east. And while, as shown as in B and C and figure 1. And the reason some bacteria do this is it allows it to travel and go to other regions. And it's hypothesized that different storms will have different types of bacteria that can form snowflakes. And as the different bacteria and the different storms move, the bacteria moves with them. So we wanted to know, and we hypothesized that the different storms would have different bacteria species present, and different storms would have different types of bacteria. So we went and took 3 different samples from Dr. O'Brien's yard. One was near her patio, one was near a table, and one was near her swing set. And we took those samples and we put them in an AeroPress membrane with a membrane filtration system. So there was filter paper on the bottom, and we squeezed the samples through that to just trap bacteria and get all the other dirt and just water out of the sample. And then we used a purifying DNA kit, the QAgen kit, to purify and lyse open the cells and extract the DNA from the bacteria. From there, we decided to performed PCR on the samples. And so PCR was performed on the 23AP, 23AT, and 23AS samples. And those samples were loaded onto a 1% agarose gel and was run on gel electrophoresis to determine the absence or presence of the 16S gene, which would be indicated on the gel with a band around 1 kb. And our results from that showed that the positive control had a band, this 16S band at 1 kb, but in all of our samples, no band was present or seen, especially at the 1 kb. So this indicates that we did not have any 16S genes present in our samples, our PCR samples, which indicates that bacteria was not present in those samples at all. So moving on from there, we took samples that were known to have 16S from prior years, and we put them on PCR. We ran them through PCR, and those samples were 23BT, 22AT, and 22AP. We also loaded them onto a gel and ran gel electrophoresis, but this time the INAQ PCR cycles were implemented to look for the INAQ gene, which would be present around 3 kb, and if we had a band around 3 kb, we should have those genes present in our samples. But as you can see in the results in this gel, we also did not have any bands around 3 kb. So this indicates that the nucleating protein that would cause the snowflake formation was also not found or present in our samples. But those samples did have bacteria present. We were not able to test objective 3, but to attempt to try to compare the abundance across the samples to determine if various species of bacteria are present, we could use Illumina sequencing, which would be beneficial because Illumina sequencing we could take our 16S gene sequences of the 3 samples in this gel, and we could run them through Illumina, and that would give us the amino acid sequences of those samples and tell us which specific types of bacteria are present and how many bacteria species are present. So right here, I just stated that the sequencing of Illumina sequencing would give us the sequences of the bacteria, information about the abundance of each bacteria in the sample, and information of whether there is variation in the bacteria in each storm. While the PCR did allow us to identify whether or not bacteria is present at all, and could give us a little bit of a hint to how much bacteria is present. Illumina could give us even more information by giving us the specific sequencing and identifying exactly which species are present. So yeah.